You guys are ready?
Yep, we're all ready.
Okay, so I thought that even though we've spoken somewhat about immunotherapy and in different venues about the immune checkpoint inhibitors, because of both the fact that they are now so solidly vested in the clinic and the fact that we are exploring not only in Mary Crowley protocols from biotechs, different protocols for our patients, but also in the fact that we are looking at the Vigil vaccine in combination with the immune checkpoint inhibitors, that it really behooves us to move somewhat beyond mechanisms of action and try to somewhat in our own minds deconstruct mechanisms of resistance to the immune checkpoint inhibitors, because clearly the more we use them and eventually for those patients that initially do not respond to them, what we call innate resistance, which actually comprises on the average about 60% of the cancer patients that we see, varying obviously within a range from tumor to tumor. So looking at both not only the mechanisms of innate resistance, why those patients don't initially respond, but also what about those patients who respond to the immune checkpoint inhibitors but then develop progression of disease? In other words, patients who've developed acquired resistance to the immune checkpoint inhibitors. And that comprises about 25% of patients who do respond to them. So that basically was the nidus for this decision to do this topic. Why don't we move to the next slide? So we've already seen this slide in my previous presentation, and I just really relabeled it because when we use the presence of TIL cells, the tumor infiltrating lymphocytes, not only the number of TILs, but also the types of TILs, in combination with the presence or absence of PD-L1 in the tumor or in the infiltrating lymphocyte. We're not really exploring mechanism. We're really looking at a predictor. It gives us a soft predictor of whether or not these patients are likely to respond to the use of the various inhibitors that we have, or whether they are likely not to respond. So what I want to do is kind of go somewhat below the surface, and rather than just looking at predictors of responsiveness or resistance, to look at the actual mechanisms underlying that resistance. So let's move on to the next slide. So the first thing that we need to keep in mind is the fact that when we look at patients with PD-1 positivity, the use in different diseases such as non-small cell lung cancer versus melanoma versus the urothelial tumors versus the squamous cell carcinomas of the head and neck, we basically know that depending upon the studies that the FDA based their approval on, that different cutoff points are used for PD-1 expression. Now, in actuality, we know that all PD-1 expression is or are not equivalent to each other. And in actuality, we can divide PD-1 expression into high versus intermediate and low. And what we, of course, look for, as we just saw with the predictor slide, in order to determine, is a tumor likely or not likely to respond to our checkpoint inhibitors, is the presence of, PD-L1 expression, but we do know that certainly in these infiltrating lymphocytes on which we, in addition to the tumor cells, measure PD-L1, we're also looking at PD-1 expression. And PD-1 expression, likewise, can be divided into high and intermediate and low expression. Now, one of the reasons, one of the ways in which these cells actually increase their expression of PD-1 is that these lymphocytes, these T cells, when they are activated, and we've spoken about activation by different antigens, and especially when they're anti-tumor by either neoantigens or what we call tumor germline antigens, that these cells will not only increase interferon gamma, but they also increase PD-1 expression. PD-1 is the receptor on the lymphocyte for the ligand PD-L1 on the tumor cell. PD-L1 can also be present on the lymphocyte as it can on other cells. But not only is PD-1 upregulated, but other checkpoint inhibitors are upregulated. And on this slide on the upper left, you can see that especially in the cells that we're most interested in looking at, the PD-1 high expressing cells, there's also a heightened increase of TIM3, which is another checkpoint inhibitor, and that increase is actually much higher than on cells that have only a PD-1 intermediate expression. So does this mean that in does this mean that in melanoma, where our cutoff point can be 1% expression of PD-L1, that if indeed the levels of PD-L1 and PD-1 have some type of parity, that in melanoma where we have a very low cutoff point, so we are including both PD-1 high and PD-1 intermediate, that the presence of an alternative inhibitor like TIM1 on the upper left-hand part of the slide, or the lower left-hand part of the slide, LAG3, another checkpoint inhibitor, could it be that the PD-1 highs also have a comparatively higher expression of these other checkpoint inhibitors compared to the PD-1 lows? So that those cells that perhaps are most susceptible to PD-1 or PD-L1 inhibition are also perhaps a little bit more likely to either have innate resistance or be able to more easily acquire resistance to PD-L1 inhibitors because of the presence of co-expressed checkpoint inhibitors such as TIM3 or LAG3. So eventually we're going to have to add to our analysis of our patients when we look for these predictors, not only PD-L1, but we need to be looking for TIM3 and LAG3 because eventually we are going to have checkpoint inhibitors for these. And these are actually already being explored in preclinical models in combination with PD-L1 and PD-1 inhibitors. So this is, certainly a possibility that can actually be overcome perhaps by combinations of these checkpoint inhibitors, although we don't yet know how much additional toxicity this may cause. We do know that the combination of an anti-PD-1 and an anti-CTLA-4 ipilimumab, knocking out two checkpoint inhibitors at the same time does increase toxicity compared to just one or the other. And how much that may be so with trying to knock out TIM3 or LAG3 remains to be seen. But this is something we need to consider. Let's move to the next slide. So reviewing another slide that I had previously discussed, but in a somewhat different light, I just wanted to review the mechanisms of immune escape in the tumor microenvironment. Now, number one, which is kind of in the middle, is reduced MHC class 1. Number two, which you can basically see on the bottom right, is the expression of FAS or TRAIL on our cytotoxic T cells, which basically results in apoptosis of these cells early on. Number three, on the upper left, we've spoken, both John and I, about the tumor immune suppressive paracrine effects coming from other tumor infiltrating cells, the Tregs, which are the CD4 FOXP3 positive, the CD8 FOX3P positive cells, the myeloid-derived stem cells, and the tumor-associated macrophages and neutrophils. Number four, the antigen-presenting cells can be dysfunctional. And then number five, when you have deregulation of immune checkpoint signaling, which we've been discussing all along. Now, one of the major reasons in cancer cells for lack of responsiveness to not only immunotherapy in general, but specifically the effectiveness of the immune checkpoint inhibitors, of all of these, winds up being number one, reduced expression of MCH class number 1. So let's move on, knowing that that is so important, the ability to be able to actually take proteins in the cell, in the cancer cell, and effectively present them to the tumor cell. So next slide. Now, this is a complicated slide, but one of the things that we all know is that cancers are heterogeneous. In other words, if you look through cancers, they're all not the same cell. The cells differ in terms of the mutated genes involved, the epigenetic changes that are in play, the different types of receptor expression in different regions of the cell. And an interesting study that was done in non-small cell lung cancer looked at 7 different regions in terms of clonality. In other words, was the cell essentially almost all one clone, or were there multiple clones? And how can that affect our responsiveness to, in this case, immune checkpoint inhibitors? So on the top, the kind of very colorful red, yellow, and blue, what we're seeing are the 7 patients that were looked at in this study. And when you look across of them, 44% of the patients studied had significant heterogeneity. And the red, let's start with the blue. The blue indicates the, essentially, if you look at these bar graphs as percentages, the blue indicates, that percentage, in which we basically have clonality. The yellow is shared expression of PD-L1. And what shared means is that there are multiple heterogeneous regions. However, most of the regions do share the expression of PD-L1. And then there are what we call private heterogeneity, where you have multiple regions, and each region is really kind of different. So there's not even a limited identity within the regions of appropriate expression. Now, the importance of this is that when we use our checkpoint inhibitors, this slide and this group and other studies supporting this basically are suggesting that for effectiveness of the immune checkpoint inhibitors, you basically need to have a low intratumoral heterogeneity of expression and a high clonality. And that if you have both of those, a low intratumoral heterogeneity, so a general uniformity of expression, and that goes, hand in hand with a high clonality, you are likely to have a much higher response to the immune checkpoint inhibitors. So if you look at that middle row labeled B, that basically shows you what happens when statistically you take these patients and you split them up by giving them different thresholds of heterogeneity. And as you can see on the right, the middle right, the top slide represents no threshold. So in other words, there's a split between the different cells within all these regions that were analyzed right at the median. So, essentially half have, higher expression and half have lower expression, based upon how much clonality there of PD1, of clonality and, how much PD1 expression there is, as a result of them all having the same clone. So they're all producing the same amount of PD-L1 rather than having subclonality where some are producing PD-L1, some are not producing PD-L1. And as you can start going down that, you can see that as you start shifting your threshold from a high threshold to a lower threshold, you move from cells that are basically homogeneous in expression to cells that, as the threshold for heterogeneity decreases, you wind up having the increased number of subclones in the group. So if you go down to row D, just move not that first square, but the second one, where essentially the threshold is 0, and that shows you that the hazard rate of recurrence in that particular group, where you have significant clonality, is 0.21. But as you decrease the threshold and you move to that third square, and then you decrease it and further and move it to the fourth square, your hazard rate keeps increasing. Your therapy is becoming less and less effective when you use an anti-PD-L1 inhibitor because you have much more heterogeneity. And as a result, you are not inhibiting as many tumor cells in a you're not inhibiting as many tumor cells in a tumor situation where you have multiple clones producing different levels of PD-L1 or no PD-L1 versus the situation where they're all producing PD-L1. So let's keep this in mind and move to the next slide while I cough. So this is just from the same paper, and this looks at what we were just discussing in a somewhat different way. It groups the proportion of tumors showing strong, weak, and negative PD-L1 expression, and the strong expression is red. And as you can see in the column on the left, with a low amount of intratumoral heterogeneity and a high clonality, especially for neoantigens, that we have strong PD-L1 expression in the majority of cells, and weak but present in the minority of cells, probably about 35%. But as you look to the right column, where you have a lot of intratumoral heterogeneity, with corresponding low neoantigenic clonality, you essentially have one patient who has strong expression, and the other patients either have weak or no expression. So one of the things we need to keep in mind about the effectiveness of not only our checkpoint inhibitors, but also targeted therapy, is the clonality of the tumor. Because as with the immune checkpoint inhibitors, tumors that have multiple regions of subclonality because of a high intratumoral heterogeneity, if you're particularly targeting one receptor, a tyrosine kinase receptor on the surface of the cell, and it's not clonal, the chances of having a significant long-term and possibly curative response is going to drop to nil if many of those clones weakly express the receptor or don't express it at all, as opposed to a tumor where all the cells are expressive of what you are attacking. So let's move to the next slide. So how do cancer cells that respond to our immune checkpoint inhibitors, and we're assuming that this is going to be due mainly to neoantigen loss, as we're now learning that neoantigens are the major antigen, the most potent antigen to elicit the immune response. And the antigen, when present, which has the highest, the cancer cells which have this neoantigens present, those with the greatest tumor mutational burden are the most likely to respond to the PD-L1 and PD-1 inhibitors. So how do we lose those effectiveness? Well, one way is that our therapy, our immune checkpoint inhibitor, is so effective that it eliminated all the neoantigen-containing tumor cells that represented the sensitive subset, and then those cells, especially when there was tumor heterogeneity and subclonal neoantigen, those cells that didn't express the major antigen that was being attacked are left. So we have the subclones now dominating, outgrowing in the tumor and representing resistance. Now, obviously, this can't occur when there are clonal neoantigens, because if all the cells are expressing the targeted neoantigen, then if our therapy is effective, they're all going to die anyway, and there's nothing to grow out. However, there is another way of acquiring resistance, and rather than just eliminating all the sensitive neoantigenic containing tumor cells, the tumor cells can acquire one or more genetic events, a deletion or a loss of heterozygosity in the tumor cells, where it's not that the immune checkpoint inhibitor is killing the cell, it's that the tumor cells are adapting to their environment by mutating, and in those mutations actually losing the initial antigen that was being targeted. Now, this type of resistance can occur clearly in subclonal neoantigens, but this can also occur in clonal neoantigens. So if you have a tumor and we're targeting neoantigen X, and you start killing cells after the first round and second round of therapy, and then you start having cells on the left beginning to mutate because, they still have this higher mutation rate because of perhaps loss of repair mechanisms, then you may be seeing adaptation by loss of the antigen that we were initially targeting. And if you lose that and there isn't a dominant antigen left, let's say in a tumor with a low mutational burden with a limited number of neoantigens, essentially, not only aren't they going to work, but even if you have, tumors that have, significantly but not completely responded, retreating them isn't going to work either. However, what you might be able to do in this situation where you have these genetic events is that if you have a tumor that has significantly responded and then it starts regrowing or recurring, in addition to the loss of antigens, you can also have had mutations that have formed new antigens. And therefore, you probably and almost definitely won't respond to the old therapy, giving back, either the same vaccine or applying the same immune checkpoint inhibitor, but you may get a response by either creating a new vaccine from the new cells, or if the new antigens were potent enough to allow for the formations of an activation of a new set of T cells, cytotoxic T cells, you may get a response out of retreatment. So the mechanism of resistance in this particular case can be very important in terms of whether or not you might expect a response to retreatment by either, as we have sometimes done with Vigil, reshaping a whole vaccine from a recurrent tumor, or perhaps retrying the immune checkpoint inhibitor in the hope that with tumor recurrence, it's not just loss of the initial potent neoantigen, but you may have had reformation of new neoantigens by new mutational events. Next slide. And this is an example that was recently published where this process has been looked at following treatment with antibodies in patients against PD-1 or PD-1 and CTLA-4. And this group was able to show that there was a definite change in mutational allele frequency, starting from baseline, which you see in that upper group with the column, the green column and the red column and the blue column. And then at the time of resistance, you essentially lost your mutational allele frequency. You lost those antigens. And by losing the antigens, you lost the effectiveness of the immune checkpoint inhibitor. So this is actually the first, one of the first published prospective evaluations of patients from baseline to resistance, responding and then not responding to anti-PD-1 or anti-PD-1 and anti-CTLA-4 therapy, going along with what we were seeing on the previous slide as a mechanism of acquired resistance. Next slide. We're now, again, going back to another way of looking at the tumor, the basis of anti-tumor immunity. And as we spoke about before, and I emphasized, there's a fairly major role of the ability of a cancer cell to be able to present antigens in an effective way to the immune system through the MHC, the HLA, but the MHC1 system, and to allow for appropriate priming of the naive CTLs. The naive TILs, and making these naive TILs into CTL, cytotoxic T cells. And, essentially, when we monitor our patients, we're looking for this process when we do the ELISpot test. Because when we start out with a negative ELISpot in our vigil treated patients, we're essentially saying, well, we have, these peripheral blood mononuclear cells, and, the ELISpot is negative. We're not really seeing activation, because when we do the ELISpot, we're looking at, the percentage of cells, the number of spots are essentially representing the percentage of cells that are making interferon gamma, which is an indication of T-cell activation. And when we treat them with vigil and we turn them on, we're basically saying, hey, these cells are turned on because they're making interferon gamma, and therefore we see a positive ELISpot test. So, one of the obvious important areas is whether or not tumor cells can do this. Can they turn the T cells on? Well, in order to turn the T cells on, they have to be able to present these antigens, especially these neoantigens, to the tumor cells. So let's go to the next slide. So before we go into that, we have to talk about how antigens are presented on the surface of the cells. And in a sense, there are three ways this can be done, and this is kind of important for us to know. There is what we call the Ubiquitin-Proteasome Pathway. Now, this pathway involves a process that takes place within a compartment within a cell, and this is true of all cells, but we're being specific about cancer cells right now, called the proteasome. And this is a process which is basically a way in which the cell can present proteins from within the cell, within the cytosol, the cytoplasm, can present some of those proteins that are not normal as antigens. And this has evolved as a way of the cell to protect itself if abnormal abnormal proteins are being made. So the body can recognize these cells as abnormal and attack them. So the cytosolic proteins that are presented by the Ubiquitin-Proteasome Pathway are abnormal regulatory proteins, proteins that are damaged or misfolded, mutated proteins, and cells that have been infected by a virus which allow that virus to use the cell to manufacture viral proteins, but now these proteins are actually within the cytoplasm of the cell. So what is particularly interesting about this Ubiquitin-Proteasome system is that these abnormal proteins will be presented by MHC class 1. Now, MHC class 1, when it presents the peptides derived from these proteins, about 9 amino acid fragments attached to MHC1 in order to activate the T cell via the TCR receptor, this almost exclusively affects CD8 cells. So lymphocytes that have the CD8 marker on them that eventually can become cytotoxic T cells. There's another pathway in the cell called the lysosome pathway. Now, this basically also will eventually create antigen presentation on the surface of the cell, but rather than presenting peptide fragments derived from internal proteins, this basically is presenting peptide fragments derived from external proteins. So how do they get these proteins? Well, by endocytosis, by sucking in, fragments of protein that are not indigenous to the cell, or by autophagy, in which it sucks in bacteria, bulk proteins, and big membrane-bound proteins These proteins come through the membrane. They're then transported, through these endosomes, which are these engulfed cell surface membrane engulfed proteins that fuse with lysosomes. And then these lysosomes are presented by the HLA system to T cells. But this group of peptides is not presented by MHC class 1. It's presented by MHC class 2. Now, MHC class 2, rather than activating CD8 cells, activates CD4 cells. Now, CD4 cells are cells that, rather than becoming cytotoxic lymphocytes, actually are more involved in the antibody-producing side of our immune system. They can also act as helper cells and help the CD8 cytotoxic lymphocytes, but these are two different ways in which the cell can present abnormal proteins. Now, there's a third way in which the external proteins can be presented by MHC class 1. And that's where a lot of our antigen-presenting cells, our dendritic cells, come in large part, because cells from the lysosomal pathway can be cross-presented through antigen-presenting cells, which include dendritic cells. And then the antigen-presenting cells essentially take the fragments from MHC, essentially that are presented by the lysosomal pathway, and they can represent them on MHC class 1. So we do have this way where we can move from lysosomal pathway MHC class 2 presentation to Ubiquitin-Proteasome Pathway MHC class 1. Whether proteins go through lysosomal or ubiquitin pathways depends both on where these proteins derive from and certain cytokines, which can actually change the lysosomal components to Ubiquitin-Proteasome components by modifying, certain proteins within these two pathways. So next slide. This is just a schematic of what happens in the Ubiquitin-Proteasome pathway, where you have an abnormal protein over on the left in the cytoplasm. It undergoes a process called ubiquitination, where it's polyubiquitinated. A series of ubiquitins are essentially just annealed to the cell. This marks the cell so that it will be incorporated into this organelle in the cell called the proteasome. And within the proteasome, it undergoes a certain amount of processing, which takes this protein and it breaks it down into peptides. In other words, you start out with this one, messed up, misfolded, abnormal protein, and then within the proteasome, it's broken down into short peptide strands made up of different lengths of amino acids. Now, this can produce optimal peptides, which are usually 8 to 9 amino acids long, or suboptimal peptides, which are, basically not the ideal size presentation of the cell surface. So it basically goes from the proteasome through a peptide transport mechanism called TAP1 and TAP2, which are the transporters of peptides involved in antigen processing, and it's brought into the endoplasmic reticulum. And in the endoplasmic reticulum, there are additional aminopeptidases which can trim some of these suboptimal peptides down to 8 or 9 amino acids in length, and then they're assembled onto presentation molecules. And then this assembled peptide presentation molecule is transported to the Golgi apparatus, and then it's transported to the surface of the cell, and that's where this MHC1 complex with the antigen, specifically the neoantigen with cancer cells, is going to be presented to the T cells surrounding it, and now has the ability to trigger the T cell receptor and activate the T cell. So this is the process that is most messed up when cells are not responding to immunotherapeutics, or indeed to the immune checkpoint inhibitors. Next slide. These are some of the mechanisms for different elements of this process to be screwed up, as it were. And as you can see, there are genetic ways in which this process can be subverted with loss of heterogeneity, involving HLA1 with different mutations, which can be involve beta-2 microglobulin, which is a component of the presentation apparatus. It can involve TAP1, TAP2. It can involve the low molecular weight proteins, LMP2, LMP7. These are parts of the immunoproteasome. As you can see, there are a large variety of demonstrated not only genetic but epigenetic abnormalities that can interfere with presentation of cells. So if you have cancer cells, and you can see over on the right, and this is only a partial list, you can see over on the right the variety of tumor cells in which these abnormalities are found. You can see why lack of effective presentation is a significant reason why cells are either not antigenic or of course, fail to respond to the immune checkpoint inhibitors. Now, just to toss it out, one of the things we've learned in our preclinical studies with Vigil, with its expression of GM-CSF, is that we do see an upregulation of MHC1. We actually also, in some cases see an upregulation of MHC2, but it is important that, given this information that we now know how important this presentation apparatus is, that the vaccine we are using has as one of its effect an upregulation of MHC1. Next slide. Now, I know John has spoken a couple of times about the fact that, another mechanism of resistance is the impact of the JAK system mutations on interferon gamma signaling. Now, this is just a slide, showing you, why that's important. Interferon gamma is, obviously necessary in order to be able to, induce the activation of T cells. So when we look at the effect of interferon gamma, in order for interferon gamma to be effective, it has to come through an interferon gamma receptor. And then when JAK1 and JAK2, these isoforms are present, that can do two things. One, JAK1 and JAK2 can cause a cell cycle arrest, but they also initiate a STAT1 mechanism which turns on an interferon gamma activated sequence, which essentially does two things. It essentially increases MHC1 antigen presentation to T cells, but it also increases PDL1 expression. So when JAK1, 2 are mutated in the bottom, you then lose the effect, interferon gamma effect, and as a result, you don't have any effect of the JAK1, JAK2 system on cell cycle arrest. So the cancer cell continues to proliferate. But also of importance is the fact that since you don't have that JAK1 STAC1 effect on the interferon gamma activated sequence, you essentially don't have an effective MHC1 presentation system. So you're not presenting antigens. And if you're not presenting antigens, and if you don't have effective incorporation of interferon gamma, well, you're also not going to have a very effective PD1, PDL1 blockade. So JAK1 and JAK2 mutations have now been shown in a number of different papers to be an indicator of non-responsiveness to the immune checkpoint inhibitors. Now, keep in mind that there are multiple JAK isoforms, and there is a JAK3 isoform. With JAK3, though, that has a totally different mechanism, and when JAK3 is mutated, it actually increases the immunogenicity of cells. So this is applying only to JAK1 and JAK2. So this is another important confirmed mechanism of resistance to the immune checkpoint inhibitors. Let's move on to the next slide. Now, again, another of my complicated slides, but what this is showing is this, we now know that in the presence of acute infection, we are able to mount an immune response to both, a new infection, and also able to mount a recall response if we've had a previous infection, let's say with that virus. Now, the response of our immune system, if we're just looking at infection right now, the response of our immune system to infection is different if it's an acute infection or a chronic infection, where you just have this long-term persistence of a virus. So when you look at models of chronic infection, and most of those have involved the lymphocytic choriomeningitis virus, we can use chronic infection as more of an indicator of what happens to the immune system when it's faced by a chronic presentation of antigens. So when you have an acute infection, you get a usual 3 or 4 day virus. The virus is in you. Your body, recognizes, certain proteins, so we mount an immune response, and, it helps us fight off the infection. Or we've had our vaccination where, basically, have a reasonable, memory response to that virus. It may take a day or two, but, that's much more quickly recalled, and we more effectively fight off the virus because we have those effective memory cells. Well, it's very different when you have this persistent, lingering, never going away virus. And that is much more similar to cancer, at least the patients we see, than an acute infection, because our patients with cancer don't have an acute problem. They have a chronic problem. This cancer is sitting there with its antigens, and it's not quickly going away. So one of the things we can do to figure out some of the immune responses in patients with cancer, a chronic problem, is to look at models that have chronic viral antigen expression, such as the LCMV chronic model. And when you look at these, what you find is that in both human and mouse models, in human chromosome 2 and mouse chromosome 1, there is a site which produces PD1. on lymphocytes. And that site, and I apologize, you have one arrow headed to site C. There should be two arrows, one headed to site C and site B. Site C and site B on human chromosome 2 bracket an open area of chromosome. So when I say an open area of chromosome, this is an area of chromosome that can actually be affected by non-structural processes such as methylation. In other words, this is now an area that is subject to epigenetic effects. So what this slide basically is showing is that in the normal situation in a chronic infection, and we are assuming that this is very possibly applicable to the cancer situation, that normally site B and site C, which bracket this regulatory area for the production of PD-1, are very heavily methylated. And when you methylate areas of open chromatin, you have the areas where you have cytosines and guanines next to each other. And there are areas, there are certain areas in chromosomes where you just have stacks and stacks and stacks of the cytidine-guanidine islands. They're called CPG islands, where cytidine is phosphorylated to guanine. And these are normally methylated because these basically normally serve an inhibitor purpose. So when you basically have a chronic infection, what you see on the bottom left, when you have a chronic infection, one significant way in which that infection is different than an acute infection, look at the four squares and look at the bottom right square where it says DNMT3A isoform 2. That stands for DNA methyltransferase isoform 2. And you can see that in acute infection on day 4, day 8, and even on the memory cells, that there's essentially no significant change in DNA methyltransferase 3A isoform 2. But when you look at the what happens as a result of chronic infection, you have a significant downregulation of DNA methyltransferase. And what does DNA methyltransferase do when it's downregulated? Well, it no longer hypermethylates these CPG islands. And if you're not hypermethylating the CPG islands in site C and site B, you are now taking off the brakes of the major enhancer, the major stimulator of PD-1. So cells that are chronically infected have epigenetic inheritable changes in site C and site B, which result in the chronic persistent upregulation of PD-1. And these cells can become so hypomethylated that there is no way that they're going to respond to anything that tries to block PD-L1. And it is important to note that this is applicable mainly to chronic infections rather than acute infections, and thus we believe much more applicable to the cancer situation, where we have chronic antigenic stimulation. Next slide. So, let's take a look at, cells that have been treated, that have been, exposed to, this chronic infection, and, have been treated with anti-PD-L1 effect. when there has been, T cell exhaustion, and that's what we call this process, T cell exhaustion. If you look all the way over on the left, that box called F, what you can see is that if you, basically have this exhausted T cell, which has just gone through the process I described a moment ago, and you treat it with an anti-PD-L1, the gray area, that gray bar, sitting at about day 25 is the period when it was treated with anti-PD-L1. You can see that in the non-treated control, there's no shift in terms of interferon gamma stimulated PD-L1 positive T cells. But in those cells treated with PD-L1 antibody, you actually have a bit of an increase, but it's a very short-lived increase, and fairly quickly the cell is no longer either effectively functioning because it's not producing interferon gamma, and also because it is not producing PD-L1. So it's essentially because it's overproducing PD-L1. So it's basically just not responding to treatment. And when you look over at the right, what you can see is that when you have immune effector cells, this is another system, immune effector cells, and you challenge it with anti-PD-L1, you actually have a significant increase. You're going from that spot on the left to the spot on the right of activation. When you look at T cells that are exhausted, and you don't do anything to them, you look at them at two different time points, you basically see that there's essentially no activation. And even if you treat it with an anti-PD-L1 over on the right with the red anti-PD-L1, you see no activation. So, essentially these exhausted cells, not only, do they not have a significant response to initial anti-PD-L1, but even when they were previously responsive and have now become, exhausted, they will have, if anything, just a meager response to anti-PD-L1. So T-cell exhaustion is clearly a significant component of our mechanisms of resistance. Go to the next slide, which is the next to last slide. And this is something that is actually coming out today. Looking not so much at interferon gamma, but looking at interferon alpha. And this group has found that interferon alpha destabilization significantly affects the tumor microenvironment immunosuppression, including, the response to the immune checkpoint inhibitors. Now, interferon alpha receptor, the reason it's important is that interferon alpha and beta go through this receptor. And when this receptor is activated, this, receptor, will allow for a significant decrease in immunosuppression and an increase in CTL survival because it is associated with IL-2 production, which is important for CTL survival. However, when you have a destabilization of this IFNAR1 receptor, as it's degraded, you basically have a significant increase in immunosuppression. There's much due to, in part, a decrease in IL-2, and you have a significant decrease in CTL survival, and obviously your CTL survival, if that is decreasing, you're losing a significant part of your immunoresponsiveness. So let's look at the last slide now. And the reason I think this new finding is so important is the fact that in order for us to fully understand, how this IFNAR can become destabilized, we have to realize that this is due in large part to communication through the Ubiquitin System. And the Ubiquitin System essentially has three components: E1, E2, and E3. E1 is an activase, E2 is a conjugase, and E3 is a ligase. And in order, for this to work as a system, it needs to be netylated by an enzyme called NED8, NEDD8. And if NED8 is present, this whole process actually supports tumor growth. So if you can, not only supports tumor growth, but it down-regulates, it deregulates IFNAR. So if you block this system, and we are actually looking at a product that blocks this, MLN4924, which I stuck in there. MLN4924 blocks NED8. Now, by blocking NED8, it essentially blocks this, what's called the Cullen ring ligase, this SCF beta trope E3, this E3 ligase, and as a result of that inhibition, everything that is a substrate of that actually accumulates. So that one effect of knocking out MLN4924 is by the buildup of all these substrates, we increase cancer cell senescence, apoptosis, autophagy, and increase inhibition of cancer cell growth, but we also significantly increase IFNAR. So that not only are we killing more cancer cells by blocking MLN4924, but we also have the potential of significantly enhancing the immune response in that situation where IFNAR has been significantly downregulated. So this mechanism and the preceding mechanism give us two eventual ways we might be able to approach subsets of patients who are not responding to the immune checkpoint inhibitors. One of them is using agents like MLN494 to block, NED8 when this IFNAR system is in play. And the other, when we were looking at methylation and epigenetic effects, is to look at ways to reverse that, such as the work being done with 5-azacitidine, which affects that entire methylation demethylation process, and which is currently being used for other reasons. But actually, when we think of it, might be able to be used if that is a significant resistance pathway to sensitize cells to the immune checkpoint inhibitors. So I just want to throw out just a whole bunch of mechanisms that we're first learning about, so that as we continue to use these agents, by understanding the mechanisms of resistance, we'll be able not only to explore, but also to be able perhaps to come up with ways of being able to make them more effective. And I'll stop there.
Thank you, Dr. Senzer.
You are welcome.
Okay, so as always, I'll send out an email. This is recorded and we'll save it as well.
So that means you have all my coughing also.
I do have all your coughing.
So you know this is not a robot.
Yes, we do know this time. Okay.